So what's the story with RNA? I mean, why is it so important? What does it do? And we already said that it's basically the love child of DNA, that DNA has given birth to RNA in a way by separating its strand and using the template strand to make the RNA with. And then we talked about the RNA making or it being made to do a specific job. And we're going to talk about what that job is, but there's actually a couple of different versions of RNA that we have to go over. First off, though, let's talk about RNA in general. We said that RNA is made by getting rid of all of these base pairs that really don't do anything, that don't deliver a message. And we said that there were things called introns and there were things called exons. And the exons meant exit, so it went out. So all of the exons are basically spliced together, and the introns are left out. So first off, what you have to realize is that RNA is a much shorter compound. It's much more compact. Everything in RNA makes sense. It's there for a reason. It's not this fluffy little molecule that's filled with all of this verbiage stuff that doesn't really do anything. So RNA is a much shorter molecule compared to DNA because of that reason. Our DNA, we said, has billions of base pairs. RNA, well, it rarely has more than 10,000. So we said that 2% of DNA is actually important, and that 2% is typically what's being copied into the RNA molecule itself. Another difference between RNA and DNA is that RNA is single-stranded. We do not have double-stranded RNA. RNA is single-stranded, and RNA comes in three forms. And all three forms are listed here in the PowerPoint for you. One is called mRNA, the other one's called rRNA, and the other one is called tRNA. The M stands for messenger, the R stands for ribosomal, and the T stands for transfer. So mRNA, rRNA, tRNA. So you know that those questions are coming to you on a test, right? One of the questions would be describe the difference between DNA and RNA, and hopefully you can do that now. You take a look at the sugars, the sugar backbones are different. You take a look at the base pairs, well, the T and the U's are different between each other. And now you take a look at length, and you understand that RNA is a much shorter compound because it leaves out all the unnecessary junk. And now you also know that RNA has three forms that we can see it in, an M, an R, and a T version. So let's talk about the mRNA first. The messenger RNA is basically a messenger that tells us what type of protein should be made. Because bottom line, that's what a lot of the RNA is made to do. It's made to go out and use this message that DNA has given it and to create a protein. And this protein is going to be made based on the messenger RNA. So it carries the message with it. It tells you what you need to make and how you need to make it. The rRNA, what we call ribosomal, well, this is the particles in which the biosynthesis of a protein take place. So you have to think of this, like I've written here, as a quote factory. So the rRNA is the factory where the protein assembly takes place. So that's the purpose of the rRNA. And the tRNA, which is called the transfer RNA, is the carrier of the amino acids. So they will go out and they will find the amino acids and they will latch onto them and they bring them back to the factory so that the factory can start assembling the protein for us. So imagine for a minute your body is a soup, and this soup has these garbage trucks that are on the inside, and the garbage trucks go out and it picks up all the trash. Well, that's the purpose of the tRNA, right? The tRNA goes out like a garbage truck and it picks up all of the trash that's out there, which is really the amino acids, and it brings the amino acids back to the factory and it allows them to go into the factory and start getting assembled into a protein that your body can then use. So there's something special about tRNA as well. The tRNA goes by what we call anticodons and codons, and we're going to have to take a look at what a codon and anticodon is and realize how this thing begins to translate a message that the DNA has just made. So here we've now kind of reached to the third point of the DNA biosynthesis type of lecture portion, and we've talked about how DNA replicates itself, and then we talked about how DNA will go through a process of separating itself and relaying a message. So this message, we were calling it transcription, and now we're getting ready to talk about how the rRNA or the RNA will go out and translate that message into a protein. So we're getting ready to go into the world of translation here. Okay, so what we're going to see in the next slide of your PowerPoint that you've downloaded is a picture of tRNA. So this is transfer RNA, and I start off with this one just because it's kind of short, it's kind of simple, we know what it does, unlike the factory RNA that comes in and the messenger RNA. We can easily look at this one here. All of the tRNAs kind of make a T. In a way, you can kind of see the T form that happens here. Let me blow that back up for a minute. Here's the T, right? There's the up and down, and there's the left and right. That's what makes the cross. So this is tRNA. Here's the 5' and 3' end, and all of the tRNAs have a very special ending, and they all end with a CCA. That is something that there's in common with all of the tRNAs that are out there. So what we see is that this tRNA is made up of these base pairs, and as we go through these base pairs, there's a specific order to them, and there's this anticodon region that's down here in this finger. And the anticodon region, if you want to look at it here in B, you're going to see the anticodon region here, and then you see the CCA ending right up here at the top. So this is just a 3D orbital of it, and this is just it kind of written out in a primary sequence. But this anticodon is very important because the anticodon region is what begins to assemble your amino acids together. So we have to look at the anticodon region, and the base pairs that are in that anticodon region actually relate and translate into amino acid assembly. So the tRNA will actually go out and look for a specific type of amino acid and bring that one into the picture. So that's the anticodon that we need to talk about. We can go through and talk about some of the reactions of how this thing does its job, but again, this is something that we're not really going to focus on at all. So we're going to leave all of that out. We don't really have to worry about that at all. Okay, so the problem is that this anticodon region, we said, starts determining amino acids, and it's made up of 3 base pairs. And whatever base pairs those are, we'll see those in a minute in a chart, but whatever base pairs those are, those relate to a specific type of amino acid. And that tRNA goes out and it finds these amino acids and it brings them back into the factory. Well, the problem is that there's 4 base pairs. Right? In RNA, we have A and U and G and C. And the A, U, G, and C, well, there's only 4, and there's anticodons that are made up of 3 bases. So there's 20 amino acids. So the problem here is that there's 64 different codons, is what we call it, anticodon and codon. There's 64 codons that are available to us, or possibilities. Now there's only 20 amino acids that are out there, right? We've talked about 20 amino acids. We memorized their structure. We talked about them 3D shapes. We've talked about their 1- and 3-letter abbreviations. We talked about how they assemble into a protein and the peptide bonds that link them up. Why did we do that? Because that whole process is getting ready to happen here, and it's all due to the RNA. So the RNA has these anticodons that translate into codons, and there's 64 possibilities. With those codons, there's only 20 amino acids, so that means some of these codons are repeats. So some of these codons will actually go in and look for maybe alanine, and then another codon set will also look for alanine. So some of these are repeats, but some of them only have one specific codon, and that's it. And methionine and tryptophan are the two amino acids that only have one codon in the discussion. So methionine and tryptophan have only one set of 3 base pairs that relate to it, and we call that codon. After all, they are some of the most least abundant amino acids in our body and in these proteins that we've discussed, and therefore they should be harder to find and not as easily accessible. And this is one of the ways that that is controlled. 3 of the 64 are what we call stop codons. So this is a signal that tells the RNA to stop the actual protein production. No more protein synthesis can happen after a stop codon. All right, so if we look at the codon chart, this is what you're going to see in your textbook. And this is a chart, and it's broken down into the different base pairs. So here we see U, G, C, and G. A and G, and then here we see U, C, A, and G, and then we have a third position of U, C, A, and G. So what I do to read this chart is that let's say that we have a codon, and that codon is reading AAA. What amino acid would that relate to? Okay, well, I go here for A, and then I go to the top one at A, and then I go here in this block for A, and I look it all up, and this is the representation of AAA. AAA stands for LYS, which is lysine. So that means if I have a codon that is AAA, then that is going to go out, that tRNA is going to go out, and it's going to find this lysine and bring lysine back to the factory so it can be used in the protein synthesis. So I bet you thought you wouldn't see amino acids again, but guess what? That's the whole purpose of DNA, because DNA gives birth to RNA. RNA goes out and it makes these proteins that your body needs, and one of the ways that it makes the proteins is using the RNA base sequences that it received from the DNA in the very beginning. So it goes out, these 3 base pairs hook up, they translate into a message, and that message is to go out and find this amino acid and bring it back to the office. And the protein assembly begins to start. Now, I put memorize up at the top. Hopefully you understand that that is a joke. I do not want you to memorize the codon table. This codon table will be given on the test. It will be given. You can use it on the homework and the quizzes if you want to. I don't care. No one has this codon chart memorized. Maybe some crazy people out there. Definitely not me. I just don't use it every day. I don't need to use it every day. If I used it every day, then that's a different story. But these 64 possibilities, who cares about them? We're not going to worry with them at all. In this chart, you're also going to see the word stop, because we talked about the stop codons that are there. And here you find one of the stop codons. So this stop codon is basically the U, and then the G, and then the A. And it has to go in that order, right? 5 prime, middle position, end. So that is the order of which you read the table. So this stop codon is going to be a U, G, A. And UGA means the production has stopped. No more amino acids will be brought in. The proton or the protein has been assembled, and now it needs to be cleaved off. So there's one of the stop codons. There's the words stop in here as well at the U position again. So this is a U and an A and an A, and then a U, and then the A again, and then the G. If you look at the C block, there's no stop codons that are there. If you look at this A block, there's no stop codons there. And if you look at the G block, there's no stop codons there either. So only the stop codons are found in the U being the first position. So that's kind of important. If U is there in the codon in the number 1 spot, that could be a signal that this protein is about to end the production. So that's a general trend that I would maybe keep an eye out for. But again, this table you can use no matter what. So that is what we call the codon table. You can take a look at how this whole thing operates if you want. This piece, this thing that looks like a nose, this is the tRNA, and the tRNA has a anticodon on the end here. And if you look, this is a U and a C and a G. And that's called the anticodon because it makes a codon. So whatever the opposite of, that's what we need to be looking for. So the opposite of U is A, the opposite of C is the G, and the opposite of G is the C. And this is the codon. And that codon is what I would find in the chart. So that's very important, okay? So when you take a look at tRNA, you need to understand that's the anticodon. And then you need to take a look at those 3 base pairs at the bottom and do their matches, their complementary matches. So U, C, and G is actually changing into an AGC for me. And then I need to find AGC in the chart. And AGC in that order, what amino acid would that bring me? So you start to see this assembly of more and more anticodons that go on. So another anticodon is coming in, sitting down on top of that. We see a match, and that has brought with it a amino acid, and here's the amino acid that the first tRNA brought, and then the second tRNA comes in, it brings another amino acid. These 2 amino acids hook up into a peptide bond. This tRNA now is done, it can leave. This is the anchor point, the second tRNA that came in. It's holding the protein now that's being assembled. A third tRNA can come in, and that third tRNA brings with it another amino acid. It matches this codon, and this codon basically tells it, okay, this is alanine. It's bringing alanine into the picture. Another peptide bond is formed, and the protein assembly continues on and on and on and on. So that is what we start to look at as far as tRNA goes. Now, the mRNA, what you need to take a look at here is that this is the messenger RNA. This is what was made by the DNA chain. So this DNA chain, that's the codon that we look for in the table. That complementary base pair brings that tRNA in, and that tRNA has with it an amino acid, and those amino acids begin to get hooked up from end to end to end. So that's the story of how mRNA, rRNA, and tRNA are all related. The mRNA is the messenger. It's bringing along the message that needs to be made, and that's coming from the DNA. The rRNA is the factory where all of this begins to happen, and the tRNA is the molecule that goes out and picks the amino acids that need to be brought into the picture and brings them in for the factory assemblage. The codon is on the mRNA that translates to an anticodon that's on the tRNA, and that anticodon goes out and it gets one particular amino acid at a time. All right, so we're almost done with the DNA lecture. Almost. But we need to continue on with that video in YouTube where we have a couple of minutes left to watch, and we need to really tie up any loose ends that we're getting ready to see here. So we're almost finished. Stay tuned.